Okay, so now that we understand something about genetics and how information is transmitted from one generation to the next, now we can talk about the process of evolutionary biology. Now, evolution simply describes change. Evolutionary biology being changes that occur in time amongst living organisms. So, many of the mechanisms of evolutionary biology might seem somewhat intuitive in the context of our modern understanding of life. But through a historical perspective, it wasn't nearly as straightforward. In fact, the idea of special creation predominated through Western society for many centuries. This being the idea that all of the organisms that we observe today on Earth are the result of a special design, that organisms were essentially created as they currently are, that they don't change through time. We know now through the scientific method and scientific process that this isn't the case, that the biodiversity that we observe amongst life is the result of hundreds of millions of years of very slow evolutionary processes. So in this chapter, we're going to go through the various mechanisms that drive that evolutionary change, and we're going to look at the key historical contributions that have led up to our current understanding of this process. So our current understanding of evolutionary biology all goes back to the scientist that you see here on the left. This is Charles Darwin. Charles Darwin was a trained naturalist. He was a biologist. And in the days of Darwin, it was not uncommon for biologists to take part on research expeditions. So there was a research vessel, the HMS Beagle, that was commissioned by the British Royal Navy to conduct hydrographic surveys of the coastline of South America. So they were constructing nautical charts showing navigational and depth information that would then be used by later expeditions and be useful for trade routes and those sorts of things. So the voyage lasted about 5 years, and each time they would make land along the coast of South America and the archipelago islands surrounding South America, like the Galapagos Islands, for example. Charles Darwin would conduct biodiversity surveys in those regions. And it was during this voyage that a number of key observations were made by Darwin that ultimately influenced his ideas surrounding evolutionary biology. So when Charles Darwin arrived back in England with his ideas, he read a book by a statistician named Malthus that described essays on human populations that described the rapid growth in human population size and the possible consequences of limited resources. That if the human population were to continue to increase in size as its current rate at that time, that eventually population size would outgrow resource availability, and there would then be intense competition amongst people for food resources and that sort of thing. And Darwin extended this idea to life in general, that not only does competition exist amongst humans in the context of an increasing population size, but that this must also be true for all other animals. That all animals, the population sizes must continue to increase exponentially until there's such intense competition for resources that the population size would then level out. There just simply are not enough resources for everyone to survive, so that only some members of the population are able to survive and pass their genes along to the next generation, whereas other individuals are not able to survive given these limitations. And so Charles Darwin's observations during the voyage of the Beagle, along with this idea that there's competition amongst individuals, led to his theory of natural selection. Natural selection basically describes differential survival and reproduction. That because there are a limited number of resources within any particular environment, that exponential population growth cannot be sustained. In other words, there's going to be intense competition amongst individuals in a population for survival. And as a result, only some individuals survive. The ones that survive are the ones that are the best suited for survival. They just happen to have the traits, the physical characteristics that make them a little bit better at survival. So whenever they survive and they reproduce, they pass those survival characteristics along to the next generation. whereas the individuals that do not survive, they don't reproduce, and therefore their poor survival traits are not passed along to the next generation. And this is the basis of evolution by natural selection. Charles Darwin compared this idea to what was known at that time or what he referred to as artificial selection. In a moment, I'm going to show you some examples of artificial selection, but this is basically where humans are capable of cultivating, of selectively breeding plants and animals, so that the progeny, that the offspring, will have more of the desired characteristics. Charles Darwin extended this to nature, that this process also occurs in nature, but the selective mechanism is differential survival. So what were the observations made by Darwin at the Galapagos Islands that were so influential to his ideas of natural selection? Well, the Galapagos Islands are an archipelago, so it's a group of islands, and they're located about 600 miles west of the coast of Ecuador. And what Darwin observed was that many of the species on these islands were very similar in appearance to species that he had observed on the Ecuadorian coast. So he asked himself the question, why is it that these species so resemble Ecuadorian species and not species of Europe or species of Africa? What's the link? What's the connection? Furthermore, he observed that on individual islands there was variation amongst populations. For example, a certain species of bird, the finch, Darwin observed that on one island the finch may have a broad beak, whereas on the other island, a nearby island, the finch may have a more narrow beak. Or on one island, the tortoise shell may be round, whereas on the other island, the tortoise shell is a little bit more flat. So why is it that these individuals resemble one another so closely, yet they are distinctly different from one another? And he was later able to answer this question through natural selection. So on the next slide, we're going to look at artificial selection, and then extend that idea toward a natural process that works by similar mechanisms, natural selection. Artificial selection is in many ways a process that has been around since the beginning of human civilization. In fact, it can be said that artificial selection is what brought humans out of the Stone Ages. Virtually all ancient civilizations practiced artificial breeding or selective breeding, whether or not they realized what they were doing. I just want you to imagine a scenario. Look at this plant to the left. This is Teosinte. This is a plant that is native to South and Central America. And you can see that this individual is pulling back some of the broad leaves of the Teosinte, exposing the grains. So these grains are of high nutritional value. In fact, they served in some of the ancient civilizations of that area as a staple food resource. So just imagine you know something about farming and you realize the nutritional value of this plant, and so you want to grow a crop of the Teosinte plant. So you grow up your Teosinte, and then toward the end of the growing season you have to collect some seeds so that next year you can grow some more plants. Well, you observe that within your population that you've grown, some of the individuals are a little bit bigger, they're a little bit more robust, and maybe they produce a few more seeds than other individuals. And so you decide, I want to plant the seeds from that big one, because next growing season I want to have all of my plants to look like this one. So you collect those seeds. The next growing season you plant those seeds and you end up with an entire crop of plants that are a little bit bigger, they're a little bit more robust, and they produce a few more seeds than the crop in the year prior. And so that year you do the same thing. You decide, I'm going to take the seeds from the biggest, most robust individual in this crop, and next year I'm going to plant those so that my crop will be even bigger and even more robust. And this is basically the process of artificial selection. You're artificially selecting for traits or for characteristics of interest so that the next progeny will have more of those traits. It turns out that this is the ancestor to corn. The ancient Mayans and Aztecs and civilizations that lived in the South and Central American regions selectively bred corn over a period of hundreds of years, and went from this teosinte, a very small stalk of these corn grains, to the very large, very robust ear of corn that we now know as corn. So artificial selection. Darwin had the idea that this process also occurs in nature as a result of differential survival. That in nature, some individuals are a little bit better at survival than others, and therefore their characteristics are the ones that are transmitted into the next generation. Just like with the corn, the ones that were a little bit bigger were the ones that were transmitted into the next generation. And over many, 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 many generations, the overall morphology, the overall phenotype, and therefore the genotype, the allele frequencies, over many, many, many generations, those allele frequencies change. One of the problems with this idea is that it must take a tremendous amount of time for evolutionary change to occur. Each new generation of individuals is not that much different than the previous generation. And so to accumulate enough small changes over a long enough period of time that the end result is a very different looking organism, this process must take millions and even hundreds of millions of years. So that was a problem for Darwin. But it turns out that around the time that Darwin was developing his ideas, a geologist named Charles Lyell published his work on the geological history of the Earth, where he was able to provide numerous lines of evidence that we live on an old Earth, that the planet has been around for hundreds of millions of years. In fact, we now know that the planet has been around for 4.6 billion years. So all of a sudden the time factor was no longer a problem for Darwin's idea, Darwin's theory of natural selection. Here's another example of artificial selection. On the top of the screen you can see a member of the mustard family. And over many years, this particular species has been artificially bred to produce a variety of vegetables that you find at the grocery store. I've shown 3 examples on the bottom of the screen here: the broccoli, Brussels sprout, and cabbage. Each of these have been bred from the same wild type. In fact, in your grocery store, most of the fruits and vegetables that you find are cultivars. They've been cultivated or artificially selected to be more robust. The reason that the fruits are so large and numerous, or whatever it may be, is because of artificial selection. The wild type tomato, for example, is about the size of a blueberry. So any tomato that you would find at the grocery store that's larger than a blueberry is the result of artificial selection and of cultivating. So this is a very similar process to natural selection. It's just that with artificial selection, humans are doing the selecting, and with natural selection, nature is doing the selection through differential survival and reproduction. So we'll now move on and talk about the concept of the gene pool. Following the contributions of Mendel with Mendelian genetics and Charles Darwin with natural selection, an emerging field formed in biology known as population genetics. So we're going to spend a little bit of time here talking about population genetics, but I first want to define and discuss a few key points. So a population is simply defined as all of the members of a particular species that live within a particular geographic region. So these members would be capable of breeding with one another, interbreeding within the population. A species is defined as individuals that are capable of reproducing with one another to produce viable, reproductively fit offspring. So if 2 individuals are capable of reproduction, we call this a species. Now it turns out that the concept of a species isn't quite so straightforward, but for the purposes of this discussion, we're going to just keep it simple. So all of the possible alleles for any given trait that exist within a population are collectively known as the gene pool. So I just want to go through a scenario using what we've learned from our discussion of Mendelian genetics and talk about the gene pool of a population. So consider the parental generation made up of 2 heterozygotes. big R little r and big R little r. So both of these individuals express the dominant trait. So if they were to reproduce, the F1 generation would be made up of 25% homozygous dominant individuals, 50% heterozygous individuals, and only 25% homozygous recessive individuals. So let's look what would happen in the F2 generation for each of the individuals in the F1 generation. So if the homozygous dominant individual were to reproduce, regardless of the genotype of their mate, 100% of their offspring would have the dominant genotype, the dominant phenotype, right? What about the two heterozygotes? What if they were to reproduce? Well, regardless of the genotype of their mate, at least 50% of their offspring would have to have the dominant phenotype. So that means that most of the alleles that are being transmitted from one generation to the next are the dominant alleles. So this begs the question, do recessive alleles decline in frequency from generation to generation within a population? Are recessive alleles ultimately driven out of a gene pool over time? To answer this question, a few scientists named Hardy and Weinberg established what is now known as the Hardy-Weinberg principle, and that question can be essentially answered mathematically through what's now known as the Hardy-Weinberg equation. So in the next slide, we're going to talk a little bit more about the Hardy-Weinberg principle and applications of the Hardy-Weinberg equation. So the answer to the question that I asked on the last slide, do allele frequencies change from generation to generation within a population? The answer to that question is no. As long as 5 conditions are met, allele frequencies do not change from generation to generation. And this was first described by Hardy-Weinberg and is known as the Hardy-Weinberg equilibrium principle. So the Hardy-Weinberg equilibrium principle basically states that allele frequencies will remain stable or will remain in equilibrium from generation to generation as long as 5 conditions are met. And those 5 conditions are that there must be no mutations within the population, there can be no gene flow into or out of the population has to be of a large, statistically significant size, mating between individuals within the population has to be random, and natural selection cannot act upon the population. Now in theory, this all sounds really good, but in practice, there are probably no populations that satisfy each of these 5 conditions. So why is this useful? Why is this significant if it can't be applied to a naturally occurring population? Well, to answer this question, think of Newton's first law. Newton's first law states that an object in motion will remain in motion unless acted upon by an external force. An object at rest will remain at rest unless acted upon by an external force. Now, in reality, in practice, we have no examples of an object that are not acted upon by external forces. Even in deep space, an object would still be subject to the effects of gravity. So why is Newton's first law significant if it doesn't actually apply to any known matter in the universe? Because it gives us a basis by which we can make measurements and observations. The same is true of the Hardy-Weinberg equilibrium principle. It provides a basis, a standpoint, a reference point by which we can measure evolutionary change, changes in allele frequencies, the amplitude, magnitude, and direction of those changes. So if any one of these 5 conditions are violated, which is the case for all naturally occurring populations, that must mean evolution is occurring. So this concept can be expressed mathematically through the equation p squared plus 2pq plus q squared is equal to 1. So on the next slide, we're gonna dissect this equation, learn what each of the variables represent, and then learn how this principle can be applied to a naturally occurring population. Now the Hardy-Weinberg equation can be used to measure 2 different things: allele frequencies within a population and genotype frequencies within a population. So let's consider a particular trait that has 2 alleles. One of those alleles would be big R and the other allele would be little r. So if you were to add all of the big R's together and all of the little r's together and combine this, then this would be equal to 100% of the alleles for that particular trait within the population. This can be expressed mathematically through P plus Q equals 1, where P are all of the dominant alleles and Q are all of the recessive alleles. So when you add all the dominant and recessive alleles together, you get 100% of the alleles for that trait. That's basically what this formula is telling us. Now, what about the genotype frequencies? If you were to add all of the homozygous dominant individuals and all of the heterozygous individuals and all of the homozygous recessive individuals, if you were to add all of them together, then that would give you 100% of the genotypes for that particular trait within a population. And this can be expressed p squared plus 2pq plus q squared is equal to 1. In other words, adding all of the different genotypes together gives you 100% of the genotypes for that particular trait. So p squared is equal to the homozygous dominant genotype, 2pq is equal to the heterozygous genotype, and q squared is equal to the homozygous recessive genotype. One useful way of remembering this, although I don't want to confuse you, so I'm reluctant to put this out there, but you can set this up in a Punnett square. If you were to cross two heterozygous individuals, the dominant individual would be R and R, so R squared. And then you would have two heterozygous individuals, big R, little r. So when you put both of those together, you get 2 big R, little r. And then when you put 2 of the little r's together, you get the homozygous recessive individual R squared. So whenever you add big R squared plus 2 big R, little r plus little r squared, you get 100% of the genotypes in the population. This is basically expressed by the formula p squared plus 2pq plus q squared equals 1. So again, p squared, the frequency of homozygous dominant, 2pq, the frequency of heterozygous individuals, and q squared, the frequency of homozygous recessive individuals. Now, I know that this might seem kind of jumbled up and a little bit confusing, but in just a moment we're going to look at an example of a population and an application of these formulas, and I think that it'll make a little bit more sense. And by the end of this discussion, you should be able to work through a similar scenario to the example that we're about to look at and determine the relative frequencies. So here's an example population. We have 800 plants in 2 different phenotypes, a yellow flower phenotype and a red flower phenotype. So we're given the information that out of the 800 plants, 20 of them have the yellow flower phenotype, and the rest, 780, have the red flower phenotype. Now, given that information alone, you should be able to calculate the percentage of alleles in this population that are dominant, the percentage of alleles in this population that are recessive, and then you should also be able to calculate the percentage of homozygous dominant, the percentage of heterozygous, and the percentage of homozygous recessive individuals within the population. So take just a moment to pause the video and, using the formulas that I just showed you, plug some numbers in and see if you can come up with the answer to each of these questions, and then I'm going to walk you through it step by step. Now, the key to answering this question is going to be in the number of homozygous recessive individuals. So we're given the scenario that we have a plant population of 800, and of those 800, only 20 are yellow. So it's fairly safe to assume that those 20 individuals must be the recessive individuals. In other words, their genotype must be little r, or whatever letter we're using for the alleles. So we can safely say that 20 out of the 800 are little r. Now, which value in the Hardy-Weinberg equation represents homozygous recessive? Recall that that is q squared. So right off the bat, given the information provided by the scenario, we can determine q squared. So q squared is equal to 20 out of 800, which is equal to 0.025. Okay, now we can determine q. So by so we have q squared is equal to 0.025. Now if we take the square root of each side, that cancels out, giving us q is equal to the square root of 0.025, which is equal to 0.16. So now we've determined what q squared is equal to, and we've determined what q is equal to. So let's go back to our formulas. So the formulas state that p squared plus 2pq plus q squared equals 1. We also learned that p plus q equals 1. Okay, so now that we've determined what q is, we can solve for p by plugging that q into this formula. So p plus q, which is 0.16, equals 1, or p equals 1 minus 0.16. So p is equal to 0.84. So now we've solved for p, we solved for q, and we have q squared. So now can we determine p squared? Yes, we can. Simply take square p. So p squared is equal to 0.84 squared, which is equal to 0.71. So now we've determined p squared, we have q squared, we have p, and we have q. The only thing left to solve for is 2pq. So 2pq is equal to 2 times p, which is 0.84, times q, which is 0.16. And if you do the math here, that comes out to 0.27. So now we've solved for all of the variables in the Hardy-Weinberg equation. So using the Hardy-Weinberg equation, we were able to determine the allele frequencies and the genome frequencies within our example population. So I've listed out all of the numbers that were calculated on the last slide here. So you can see in terms of allele frequencies, 84% of the alleles in our sample population are dominant. 16% of the alleles in our sample population are recessive. In terms of genotype frequencies, 71% of the genotypes in our population are homozygous dominant, 2% of the genotypes in our population are homozygous recessive, and 27% of the genotypes in our population are heterozygous. So with the information that was provided using the Hardy-Weinberg equation, we were able to determine both the allele frequencies and the genotype frequencies. Now I encourage you to read through this scenario again and try to calculate each of these numbers on your own so you can really develop a working knowledge of how the Hardy-Weinberg equation is applied. I'm going to give you another scenario on the next slide, and this will just be kind of up to you to work through and try to come up with a solution. Now on an exam, I'm not going to ask you to do any math, so you're not going to have a calculator, but I will ask you specific questions about the Hardy-Weinberg equation where if you can work through each of these steps, then you'll definitely be able to answer any questions that I'll throw at you. So this would be a really good way to develop a working understanding of the Hardy-Weinberg equation, and I encourage that. So why is this significant? Well, if we were to measure the allele frequencies of a population and then go back a few years later and measure the allele frequencies of that population again, what will be observed? Would they be the exact same, or would the genotype and allele frequencies be different? Well, if any one of those 5 factors is violated, then the genotype frequencies are going to change. In other words, evolution is occurring. So with the Hardy-Weinberg equation, we can measure the rate, the magnitude, and the direction of evolution for a particular population. So the Hardy-Weinberg equilibrium principle is fundamental to the study of population genetics. So here's another practice scenario, set up basically the same way. So take a moment, you can go ahead and pause it here and take a moment to work through this, apply what you've learned about the Hardy-Weinberg equilibrium principle, and see if you can determine the allele frequencies and the genotype frequencies. And I'll include the solutions to this problem in the description.